Okay, kids, we've covered a lot over the past few weeks, so I hope this doesn't drag on too long. Okay, so the first thing we covered was DNA technology, and mostly what we're talking about is this process called transformation, which is very simply getting this small piece of circular DNA called a plasmid into a bacterial cell that didn't have it before. And there's a whole process for that, and we'll talk about the actual protocol on the next slide. The purpose of this is we use these plasmids for constructing any DNA construct that we want to use in modern biology. We use them for lots of things. Could be expressing our favorite protein in a particular tissue. Once we purify the DNA and give it to another animal. It could be making proteins like insulin in vitro, things like that. But a little background first. Plasmids are actually one of the means that bacteria use to communicate sexually because they don't actually exchange DNA another way. They can have useful things in these plasmids, such as resistance to antibiotics is the big one, and also extremely relevant to our process. So, as represented here, this E. coli cell is not capable of resisting an antibiotic, such as ampicillin. But if it comes into contact and absorbs this plasmid, which contains a gene which gives resistance to ampicillin, it can then resist ampicillin and grow fine in the presence of ampicillin. So this is how we actually experimentally make the bacteria accept the foreign DNA. So what we do is we take a fresh colony of DH5 alpha E. coli. DH5 alpha is a specific strain. It has a lot of advantages. It grows great, divides quick, doesn't kill anybody if it gets out into the water supply, and it can be transformed, which is of course why we're using it for transformation. So to do that, we're going to suspend it. You can suspend it in water first, but really what we did was suspend it just in cold calcium chloride, and we let it sit there for 15 minutes. The exact mechanism of all this is a little sketchy, but calcium chloride is thought to permeabilize the cell wall, cell membrane of the E. coli, as well as actually to make the DNA stick to the membrane. So when we heat the bacterial cell up to 42 degrees Celsius, that's a little bit warmer than body temperature. We do that for 2 minutes, and that theoretically shakes up the outer membrane enough that it starts to kind of make endosomes and start absorbing little pieces from the membrane, and therefore incorporate the plasmid DNA that's stuck to the membrane. In order to get things sealed back up, since we kind of burst the membrane, we quickly chill it after those 2 minutes. Then we let it sit at room temperature, or 37 degrees, which is body temperature, which is where E. coli lives. We got it from humans. That's our body temperature. And we do that for several reasons. It can be as simple as just letting the E. coli recover. It can give it a chance to actually express whatever genes in the plasmid that we just gave it. So in this case, we're giving it an ampicillin resistance gene. We're letting it ramp up production of the gene product of that. I forget the name of it right now, beta-galactamase or something like that. And also, if we don't think our transformation is working great, the extra time at 37 degrees gives you a chance for the cells to divide and then have more colony-forming units. So after we have that, we're going to plate them on the appropriate medium. And what that means is we're going to put them on a petri dish that has within it LB, which is a nutrient broth, mixed with agar, not agarose, agar, and that'll just gelatinize it and let the bacteria grow on the surface. So the results of that, well, this is the experiment we did, and it's got several controls within it. So we have two positive controls for growth. So if our bacteria were healthy enough to actually grow, if you put them on this LB plus agar plate without anything to impede them. So no ampicillin, that's this one and this one, the first and third ones. If you do that, then they'll grow like gangbusters, and within one night they'll have covered the plate completely. However, if you didn't actually give it plasmid DNA, so that's this vial here, which is minus amp resistance, if we didn't give it DNA, we just gave it water, and you expose it to a plate that has ampicillin in the agar, nothing will be able to grow. So the ampicillin will kill off those bacteria, or at least impede their growth. However, if you had a successful transformation, so we gave it DNA, we went through our whole protocol, and then you played it on a plate that has ampicillin in the medium, it will be able, only those cells that actually took up the DNA will be able to grow. And what they do is they form these little colonies of, tens of thousands or millions of cells all around the same place, all the same genetic identity, especially all containing that plasmid. Okay, the other thing we covered was restriction enzymes, and ultimately what these are is a way to cut DNA in a very specific place. And what they do is they cut these, enzymatically, they cut these palindromatic sequences. They're very specific within a DNA sequence. So what do we mean palindromatic? So it reads the same going forward and backward. So in this top example here we have GAATTC, whereas at the bottom it's GAATTC. So no matter which way you read the DNA, and you always read DNA 5' to 3', it will be the same. When the enzyme actually cuts, it doesn't cut it clear across like this. It cuts it with little overhangs left, and that helps us cut and paste them back together later when we've got our when we're making whatever DNA construct we want to put together. What we learned about mostly, though, was the end where we're cutting pieces of DNA and kind of analyzing how many sequences are in that piece of DNA that can be cut by a specific restriction enzyme, like EcoR1. Okay, so this is the specific vector that we cut. Vector is the same thing as a plasmid. Again, it's circular. It has a known length, 3,200 base pairs. And what this map is showing is where various enzymes cut. So EcoR1's up here. I believe on this map the SAC1 site isn't shown, but it's somewhere about like this. So if you make both of those cuts, a cut here, a cut here, what you end up with is two linear pieces of DNA. Given that we know where they are, we have a predictable size from those two. Excuse me. So after we've made our cuts, what we do is we run them out on an agarose, not agar like we had in the LB plates, agarose. Run them on a gel. So DNA is negatively charged. We load it up here in these wells. We apply a voltage to it. It runs toward the positive end, which is down here, where smaller pieces migrate quicker than bigger pieces. So this was some of the results we got back from our digest. And what we did is we ran uncut DNA, we ran cut with Hindi3, we ran with SCA1, and then we ran with both cuts, and then we ran a DNA ladder or a marker. So there's a couple of things that happen with this. When you have a single cut, you linearize the DNA and it runs at a given size. These two are slightly shifted. It's probably just an anomaly of the gel. Whereas when we cut with both, remember we had two pieces. So there's a slightly bigger piece, which ran slower, and a slightly smaller piece, which ran a bit faster. And then there's some strange things that happen with uncut DNA. I'll talk about those in a minute. The purpose of running along the gel, though, excuse me, running a ladder with the gel is so that we can tell what size our fragments are. So we also know what size the fragments are from the DNA ladder. And so this one was kind of a strange, it's a bit older ladder, but it has given sizes, let's see, of 564 base pairs, 2,000 base pairs, 2,300 base pairs, 4,300 base pairs, and so forth. And those correspond to the bands we see here. So the quickest running is the 564, next the 2027, then it is very close to the 2322, then the 4,000, and so forth. We do this so that we can determine the size of a band that is unknown. So in order, if it was if an unknown band was right next to one of the DNA ladder marks, we would know that it's the size of the ladder mark. However, if it's in between, like these guys are, or these guys are, we have to kind of plot our knowns so that we can see where the unknowns compare against them. So what we're going to do is we're going to actually measure the millimeters that it traveled. So in this case, it traveled, 19 millimeters. This one it traveled 24 millimeters, and so forth. And we assign that. Actually, we recorded the difference. So 18, 23, 26. We take those values and we plot them, and we also measure how far our unknowns traveled, so that we can kind of. So when we plot these, we see something like this, and they're plotted on a log scale, actually. So this y-axis is logarithmic; it goes up by 10 over a linear time, whereas this is just the linear distance, the linear scale of the distance that it migrated. So again, the one that migrated the most, something like 55 millimeters, corresponds to 564 base pairs. These are probably our 2,000 bands, yes. So this migrated 38 millimeters. This migrated 40 millimeters, and those correspond to the 2,300 and the 2027 base pairs. So when we plot it like this, if we have an unknown that rests somewhere in between, we can actually just use our grid to figure out where what size it is. So in the case of, say, these two unknown bands, we measure the distance that they traveled, and say we do this one, the faster running one, and we say that it is 44 millimeters. So we just count off. That should be 42, 44, trace up to where it hits, and it should be somewhere in the 1,500 base pair range, because it's between 1,000 and 2,000. So that's how you're going to do that. Like I said, there's some funny things that happen with uncut DNA. So if this is all the same vector, same plasmid cut with restriction enzymes, but with the same restriction enzyme. And so here in the first lane, that's just the linear form. However, if you don't cut it at all, you tend to see two bands, because two different things happen. There can be supercoiling, and that's the stuff that runs much faster. So the DNA molecule actually appears smaller than it is because of the way the DNA is twisted. Then you can kind of have it where it twists on itself, but it kind of backfires and just kind of makes the molecule a little bit bigger. Certainly the supercoiled is more common for circular plasmids. Then if you do a partial digestion, that's shown in lane 3, you'll still have some supercoiled, you'll have some that actually linearized, and then you'll have some that's nicked, where it only cut one strand of the two-stranded DNA. And that actually makes it run slower. Okay, so next we had a review of tissues. I'll have you remember that we discussed tissues as being part of one of 4 groups: connective, epithelial, muscle, nervous tissue. I think this is a good place to mention that the different cancers are named for the different kinds of tissues that they come from. So connective tissues are going to give rise, let's see if I can get this right. Connective tissues are going to give rise to sarcomas. Epithelial tissues are going to give rise to carcinomas. Nervous tissues, if anything, are going to give rise to blastomas. They're going to be derived from kind of the progenitor cells that exist in the nervous tissues. And then there's another set, there's not really anything in particular for muscles, but there's another set for germ cell cancers and then blood cancers, leukemias, and lymphomas. So these all have different characteristics. Hopefully you remember our discussion of them. Epithelial tissues have those nice cuboidal shapes. They form a free surface, meaning they have one side that's attached to something and one side that's free to do something else, like absorb nutrients. Connective tissues like to extrude a lot of extracellular material and make things stick together. So these guys are really big on cytoskeletal elements and extracellular matrix or collagen. Muscles have the ability to contract, and nervous tissues have the ability to propagate action potentials and serves communication. Okay, so the first tissue that we looked at that was nice and complex was the skin. There's many different layers that we could observe. Down below the skin, there's a level of fat for insulation and cushioning. Somewhere in between those are the very specialized structures. So the sweat glands and the hair follicles. Attached to those are the sebaceous glands that actually extrude the oil that keeps your skin from drying out. Most of the body of that is through the dermis. That's this layer of connective tissue, blood vessels, and so forth underneath. Then we get to the part of the skin that does the bulk of the work that we think of as skin. That's the epidermis. Epidermis has a layer of epithelia called the basal layer, and if you see an actual histological section, that's here. You can note the nice connected arrangement of the epithelial cells along that border. These projections of the dermis up into it are called the papillae. I'll have to check that for myself. But anyway, so our epidermal epithelium, the basal layer, is going to proliferate, and then as the cells move toward the outside, they're going to gradually become what they're called granule cells. But what's essentially going to happen is they'll become more and more likely to express high levels of keratin, which eventually gets to the point where it breaks down, pushes apart the nucleus. They become enucleated, they lose their cytoplasm, and that's all happening. That's happening in earnest once it gets up to here. Then by the time they're here, they're missing their nucleus, and by here they're pretty much dried out and keratinized. So the keratin in these cells cross-links to each other to make the skin waterproof in combination with the oil that gets trapped in there. So again, this outer layer is keratinized, and it's mostly just a big chunk of protein waiting to be kind of flaked off as your skin wears. So we also had a review of the frog intestine. I just want to remind you what those structures look like. First off, we have the space in the middle called the lumen. Then we have a series of epithelial cells deeply invaginated between them to provide a high level of surface area, so that when nutrients pass over this area of the intestine, they have a high surface area to absorb nutrients. Sorry if that was doublespeak. Underneath that, we have the supportive tissues. That's also called the mucosa, as you can see here. Underneath that is the supportive and connective tissues, blood vessels, various extracellular components making things sticky. And that's called the submucosa. And then outside of that, we have two layers of muscle. I forget which is actually which one's more interior than the other, but there's a longitudinal and a circular. I apologize, but there's two layers of muscle in there. One's for constricting the intestine, one's for making it squish along its long axis. So that gives you the different movements of the intestine. And then on the outside, there's a coating, a single squamous epithelia called the serosa, and that's to protect the organ. So we had a look at several connective tissues. The examples are shown here. They're in order according to the text, and I won't go over that. But again, they're famous for excreting extracellular matrix and various connective fibers. You can see that here. So there are several specializations upon this. Okay, we'll go over it. So there's muscle, there's fat for storage, there's bone, obviously leaves very strong residue, and actually blood is considered connective tissue, both because of its developmental origins as well as its function of connecting the different tissues of the body. So they communicate the oxygen to the other cells, so that's why they're considered connective. At this time, I'll point out the red blood cells in particular as one of the tissues that we looked at. So the red blood cells, the erythrocytes, are here. You'll notice they have a typical dimple shape, and they're interspersed with these big old cells that have stained positive for what is actually a nuclear dye. So the key thing to remember here is red blood cells don't have nuclei. They've already fulfilled their gene expression purposes, and throughout their roughly 3-month life, they won't need to make anything else. And these cells that are interspersed with them are white blood cells, or leukocytes, and those do still have their DNA, and that's why they stand out when stained with nuclear dye. We looked at 3 different muscle types. They each have their own characteristics. Skeletal muscle and heart muscle, cardiac muscle, both have striations of actin and myosin, I believe. You guys know better than me, it turns out. We knew in class. Key thing about skeletal muscle is, again, the striations give it away, and I have 2 different pictures up here. If you blow up this one, or if you look really hard, you can actually see the striations. They're also multinucleated, so they have multiple nuclei in each fiber. That's a function of their development. The other thing about these is their function. This is the actual locomotion of the body, and they're the only voluntary muscles. So cardiac muscle and smooth muscle beat on their own. Cardiac muscle does it all in sync, and the reason that they can do that is because they have these series of gap junctions called intercalated discs. This allows communication of kind of the ion gradients within the cell, so that when one decides to fire or contract, they all do. That way they work in a unit, because you want your heart beating as one continuous pulse, not one discrete pulse, not one continuous activity of a few cells firing. And so then here's 2 different pictures where you can see the intercalated discs between them. And smooth muscle has a typical spindle shape. That means it's pointing on the ends. Its arrangement of actin and myosin is different. It's cross-hatched across each. This is what it looks like. It's not multinucleated, and it's primarily used in tubes, so either in the lining of blood vessels or in the digestive system. Those, you'll remember, are not voluntary contractions. You can't decide when you want to move your bowels. So that's what those. And then finally, we looked at the spinal cord. And this is a little bit different stain than what we saw, but it reveals the structures a little bit more starkly. And what we could see, there's an internal canal that communicates the cerebrospinal fluid. Then there's a gray matter, that's this butterfly shape here. And then there's the white matter outside of it. The white matter is named white because when you open up these tissues, it actually looks white because of all the fat that's in there from the myelin cells. So myelin cells insulate the axons as they run north to south, and this boxed area we'll zoom in on, and you can actually see both the cell bodies residing in the gray matter, that's where they live, as well as just the fibers running on the outside. So here we see that. We see neurons are very much the biggest cells in this. They have typical shape, the axons and dendrites coming off them. In this case, we'll really just see the axon. But again, these guys are much bigger. And here, as you get out into the white matter, you can kind of pick any one of these dots. So let's say this guy. In the middle is the axon of a neuron whose cell body lives, we don't know where, could be 3 meters away. Well, maybe not in a human, but anyway, it is then wrapped round and round and round and round and round and round by a myelin sheath cell, which in cross-section here it looks like a vacant space, but it's essentially just a collection of fat to insulate the electrical signal running through the plane of our screen back toward the brain or down toward where the signal needs to go. I think this is the standard picture we had for our neurons, so this gives a better shape of the perikaryon. That's the bulk of the cell body of the neuron. And it also gives you a sense of the scale of what a neuron looks like compared to a glial cell. So they are much bigger. Glial cells, often overlooked, comprise about 90% of our nervous tissue. They have important functions in maintaining neurons. They may actually have some signaling capacity, but they are definitely very important for maintaining the environment for neurons. So they nourish, they contact the blood vessels, so they communicate the oxygen. They mop up leftover neurotransmitters and just in general make a nice environment. Having spent a lot of my work on this, I'm just going to put a cheap plug in. They are actually fascinating cells, and I'm just going to give you one picture to show you that. But if you do a Google Images search, you'll see some really fantastic things from glia. So this is one example of a typical astrocyte that's not even as complex as it will eventually become on a cell biology level. Okay, next we started our anatomy sections, and we did this on the fetal pig. I will remind you which direction is which. Anteriors to the front, posteriors to the butt. Dorsals on the top, think of a dorsal fin on a dolphin or a shark, and then ventrals on the belly. When we talk about left or right, we're talking about the pig's left or right. So normally, because he's on his back, that will be opposite for what we conceive of left and right. Remember, we looked at them first from the outside, and we tried to identify and compare to our own, identify what we could and compare to our own physiology, our own anatomy, sorry. Especially concerning the joints, they walk on their toes, and their elbow and their wrist are at seemingly different places than ours. Then their shoulders will be back in here. So don't be tricked up by the way and the where that these things bend. We also have a couple different names for things. External nares for the nose, auricle. There's actually another term that I'm forgetting just at the moment. Look up your lab PowerPoints for that, for the ears. And then we could tell if it's a boy or if it's a girl from what we see on the outside too. So the mammary papillae are a good indicator that it's a female. It's not absolute. If we have a look at the back, we can definitely see this. So there's an actual, there's another papillae for the female and not for the male. He'll also have the protrusion of his scrotal sac, that's where the testicles are going to come in. And we'll see in the next couple slides, she'll have an actual opening at the back where he'll just have the protrusion again. So on our lady, we obviously have the butt, but she's also got an extra opening for her sexual organs. in contrast to the guy on the next slide. He's got the same butt, but he's actually got a protrusion here instead of a hole. I'll point out that the female was actually kind of evaginating, so her insides were kind of coming out. So don't be confused. Okay, so to discuss the anatomy, I'm going to trace three different things moving through the body. I'm going to start with food, later we'll do air and blood. So food is going to start in the mouth, and so we have our various kind of duh structures for this. Tongue's going to be important for swallowing, obviously going to have teeth, as well as the hard palate and the soft palate, also used in kind of mushing up food. So once we start thinking about going down the esophagus, we're going to encounter this general area in the back called the pharynx, and that's essentially that's just the hole, but it's going to be busted up for its different functions. So when you're toward the mouth side, that's the oropharynx. Once you get actually right in here, you're actually connecting with the sinuses and the breathing tubes. So that's the nasopharynx, nasal. And so that part of it is important for breathing. But if you kind of go past that and then back down, you're in the laryngopharynx, which basically just puts you toward the south side of the pig. What else here? Epiglottis is of course important for deciding which way, whatever you're taking in, whether it's air or food, which way it goes. So that's the decision between windpipe and food pipe. Again, we call the nostrils nares for our pig, but I should have gotten to the nares later. Okay. So once we've decided to swallow, we're going to go past a series of sphincters, and I'll kind of review them if I forget them at the end. But we're going to go down the esophagus. There's a couple of sphincters which act as gateways there, and that's the only purpose of this slide, is to see the esophagus. We're going to trace that all the way down to the stomach, and we'll have a better picture. Okay, so this is kind of how the organs exist in situ. We're going to disturb these so that we can get a better look at them, but we should be able to name all these structures. Liver, important for digestion. Stomach, important for digestion. Spleen is actually used for cleaning up the blood supply and actually has immune function. So it's kind of a host for immune cells, white blood cells. Large intestines, small intestines, obviously going to be doing most of the nutrient absorption. So let's push those aside now that we see, and let's trace the actual movement of the food. So we're going to come down from the esophagus. Let's see if we can actually see the entry point on it. No. We're going to come down through the esophagus into the stomach. And there's going to be a sphincter gating that, the cardiac sphincter or the esophageal sphincter. And what that's going to do is it's going to open, let the food in, and it's going to close, keep the food in. I must have said that wrong. It's going to open, let the food in, but it's going to stay closed to keep the food in. Maybe I said it right in the first place. Anyway, stomach has ridges inside. It's going to facilitate the mushing up of the food. You should remember, have high levels of acid in here that's going to start breaking all the different bonds that we need to break. Some of the sugar bonds, definitely peptide bonds. Anything that we need to break up into small pieces is happening here in the stomach. So once we've completed that function, we're going to move out. We're going to go through this very thick sphincter here called the pyloric sphincter. So that again is a circular muscle that's gating entry into the next structure. And everybody's going to meet up in this first part of the intestine called the duodenum. Terms here. So many different things are going to meet up in the duodenum. We have to trace them all. So the liver is actually producing bile. Bile is essentially a surfactant. It's going to help us absorb the fats and lipids that we need. So as it produces that bile, it's going to be stored in the gallbladder. Gallbladder has its own duct that connects to the duodenum, and so that's how the bile salts get in there. Pancreas also has a digestive function in producing bicarbonate and other digestive enzymes in addition to that. Those are created via the pancreatic duct to the duodenum. They all meet in there. The bicarbonate that it's producing is actually used to neutralize all the acid we just had from the stomach so that it doesn't wreak havoc all the way down the digestive system. I should pause here and remind you that if I didn't say it, the esophageal sphincter, the cardiac sphincter, is what's preventing acid reflux from burning up from the acid in the stomach reaching up into the esophagus and damaging that structure. So that's another thing that can go wrong and create a disease state. Okay, so we've got a meeting of many different things: the food, the chyme, bile salts, and pancreatic enzymes and bicarbonate, all in the duodenum. We're going to get all together and we're going to start moving through the small intestines. Small intestine is going to be responsible for the bulk of the surface area we're going to pass this food over, and so therefore it's going to be responsible for most of the nutrient absorption. So as it comes across the epithelia we discussed before, it's going to start leaching off everything that our body needs and passing on the junk. Eventually it will get to a point, I think we have a separate slide for this, where the small intestine and the large intestine are going to meet. There's several different structures we can point out there, but really the most important is going to be the cecum, and there's another valve there, the ileocecal sphincter, that's going to hold up passage until it wants to move the chyme forward. I think we'll just so I'll discuss that in the separate slide. But then most of the nutrient absorption has actually been accomplished. When we move to the large intestine, the food's basically going to be just dehydrating and getting ready for excretion. If you're going to take in food, you might as well keep the water that goes with it. Yeah, okay. So here at the junction between the small intestine and the large intestine, so it kind of flows up like this and hits here. The cecum is this structure. It's analogous to the appendix in humans. For pigs and herbivores, it's an important source for storing bacteria that will actually contribute to the digestion of cellulose or just of beneficial bacteria in general. And the appendix kind of conserves this function in us. We think it's a reservoir of good bacteria, things that, say, make vitamin K. So as things are, so the chyme will pass over this structure, they'll kind of get inoculated with these good bacteria, and then they'll kind of rest in the large intestine, again, while they dehydrate. The body absorbs the water from the food and moves down toward excretion. And then finally we'll come out, and I don't think we have a slide. I think we covered most of these sphincters. There are various other, I've forgotten what the odi were at this point, but there's also, just remember, sphincters are important as valves. So if you're busy holding it and you really have to go to the bathroom, you're also encouraging another sphincter to stay closed. Okay. Hopefully I'll have a much more succinct description of the movement of air. So again, we're starting at the head. Pig like us can breathe through its mouth if it wants to, but mostly we're going to be concerned about moving air through the nostrils or nares. They're going to pass along the nasal cavity and sinuses. Remember, we chopped those up. We could see the high surface area used for trapping dust and bacteria and everything else. As well as for kind of hydrating the air as it goes into the lungs so that we don't dry out. Again, those passageways come back here. They're going to meet up with the pharynx area, connect with the nasopharynx subportion of that, and start going down the windpipe. So once down the windpipe, that's the whole trachea. There's an enlargement called the larynx. That's where our vocal folds are going to live. Vocal folds also called the glottis. In human males, this gets pronounced into the Adam's apple. And to address a little controversy up in my class, women do have this structure. It's just not rearranged the way that it is in men during puberty. Okay, so we're going to trace the air. Air's going to flow all the way down trachea into the main branches called the bronchi. This one is obscure, but you can see the opening of this one, which go into the lungs. We have 5 lobes, same as in pigs, only 2 on the side of the heart because the heart's bigger, and kind of obstructs making a bigger lung structure. Inside, we're going to go to cartoon now. We have a series of finer and finer pipes that the air is going to be traveling through. So we got bronchus, plural bronchi, again, finer and finer air tubes. But the point we really want to make is the actual gas exchange only happens in these small functional units called alveoli. You can see that blood vessels are wrapped around the alveoli, and that's a function of how they exchange the gas. Let's see. So then there's one more important structure for breathing, and that's the diaphragm. There are various muscles involved, both in drawing in air and drawing out the air. But remember, we breathe through something called negative pressure, and that means when we move this diaphragm muscle to the posterior, when we breathe in, okay, do this with yourself and kind of figure it out. This actually expands the chest cavity such that there's negative pressure in this sealed container that is our chest. That makes the lung bigger, that makes the air pressure inside of it lower, and makes air draw in so long as any of your openings are open. To breathe out, mostly we just relax the diaphragm, let it come back in. Because this is negative pressure, you need an intact chest cavity to be able to exert the pressure on the lungs, the negative pressure on the lungs. Okay, and this is just a picture of the diaphragm. Remember, that was connected at the base of the ribs, and this poor guy's had most of his ribs cut away. Okay, blood. So we didn't yet get into the complex net of blood vessels, but what we want you to be able to do is trace the flow of oxygenated and deoxygenated blood through the heart. And we have what's called a double circulation system. That means we have 2 different pumps. One pump goes to the lungs, that's represented here. One pump goes to the rest of the body. Okay, and we want you to know the different vessels that are important for all those different things. And I think I'll go through it, some of the basics twice, once on this figure, once on the next. So, again, remember when we talk about anatomy, it's the individuals left, right, not the observers. So in this case, we're looking at a ventral view of the heart. So we're looking at it from the belly, and that means that the left side of the individual is over here and the right side is over here. So if we start first with deoxygenated blood, it's going to be returning from the system, and it's blue because deoxygenated blood has a blue appearance. You can see that in the veins of your body. They're going to enter the right atrium. Right atrium. Atrium is a term for an entryway, so we can think of it that way. Veins are going to be leading up to this. The blood flow through them is regulated by actual contraction of skeletal muscles. So it's actually you moving your legs that gets your legs to wake back up and get the blood back out of them. Anyway, there's two major blood vessels supplying this. The superior because it's on top, inferior because it's on the bottom, vena cava. Okay, so once the blood makes it into the atrium, it's going to pass through the atrioventricular valve. In this case, the tricuspid valve is another name for that. The first of these that goes from the atrium to the ventricle is tricuspid. We'll see another one over here, again on the left side, but that one is the bicuspid. Okay, once we make it into the right ventricle, through the blood squirting through this valve, we now have the major muscles in the ventricle going to be doing some of the heavy lifting. So they're going to put, when the left ventricle constricts, the various pressures and structures of the valves are going to make this one close and this one open, and the blood is going to squirt through the pulmonary artery. Despite this being blue, it is an artery. It's an artery because it has the muscles to maintain a certain amount of pressure to get them to the lungs. We need that because the lungs contain this fine mesh of small capillaries that's hard to get blood actually through. So we need a lot of pressure here. Once it makes it to the lungs, obviously their function is to reoxygenate and decarbonate, I guess? I don't know, to remove carbon dioxide. They do that all at the same time. And it's going to come back through the pulmonary vein to the heart. Then it's going to enter the left atrium. From the left atrium, it's going to go through the tricuspid. I got it wrong. It's going to go through the bicuspid valve, also called the mitral valve. It's just going to pause here in the atrium, and it's going to be squirted into the left ventricle, and this is where the main pumping of the body is going to be done. Left ventricle is the biggest structure. That's the part you can feel through your chest, and when that contracts, it's actually going to be pushing the blood system-wide. So it's going to go out to all the different structures that need blood. In case I forget on the next slide, I'll remind you that the atria beat at the same time and the ventricles beat at the same time. And that's a function of their function. This is general vocabulary. It would be good for you to know. I encourage you to know, of course, all the different chambers, some of the landmarks. The smaller arteries I don't think are as important, but definitely need to know vena cava, the aorta. Actually, that's something I neglected to say in the last one. So the final pump is going to go out through the aorta of the oxygenated blood. The membranes cut away, and then the pulmonary arteries up here. Okay, so we're going to trace the blood one more time. Again, we're going to come through the inferior and the superior vena cava into the right atrium, the loading place of the blood. Again, we're blue because we're deoxygenated at this point. We've come from the body. It's used up all the oxygen in this blood. Right atrium, left atrium, they both contract synchronously, but again, we're only concerned about the right atrium. It's going to flow through the tricuspid valve into the right ventricle. Right ventricle is a pretty big structure because it has the pretty big job of pushing blood through the heart, through the lungs. Lungs again have all those smaller structures that's kind of hard to actually move fluid through. When it does that, remember that valves are important. We need the blood to actually flow to the correct place. That means we need the tricuspid valve to close tightly. The way that it does that, I made the analogy in my class to a parachute. So if there's a parachute, the air is pushing against the folds of the parachute itself and filling those up and creating tension, while the cords actually keep the tension. So the same thing happens in the heart. You have chordae tendineae attached to the valves. When the blood starts to be moved around by the contraction, it starts to fill up the folds of this, not a parachute, but a valve. And then the chordae tendineae actually hold it in place, keep the tension, and keep it closed instead of blowing all the way through. If it blows all the way through, obviously you're not moving blood through the heart efficiently, and you could, conditions like this may be a heart murmur, and you're going to have problems, living, because you're going to need to be moving blood efficiently to actually oxygenate the tissues that need it. Okay, anyway, assuming things are working correctly, the blood will run into this valve and not go through. It'll instead go through the pulmonary valve once the pressure is greater than the pressure in the pulmonary artery. Pulmonary artery, again, is an artery, not a vein. It needs the tension provided by the muscles in the artery to actually get it to and through the lung. So it's going to go out this way, get oxygenated by the lung, come back red through the pulmonary veins. That's more of a passive process. That's why we only need a vein. That blood will be loaded into the left atrium. Once that contracts, it will push the blood into the left ventricle, and this is going to be the main pumping station for the whole system. So again, valves are important. When the left ventricle, sorry, I meant to point out this is also the point where you, this is bigger, and it's the part where you can actually feel with your hand your heart beating underneath your ribs. So when this contracts, we have the mitral or bicuspid valve sealing off backflow to the left atrium, and instead letting the blood flow out through the aorta to the various tissues that need blood. If you have a valve defect, this is probably the more serious one, one that will get you into trouble immediately, because you can still have negative pressure pulling blood through the system this way, but if you're not actually forcing the blood out to the other tissues, you're going to be fainting and suffocating before too long. One last note, I think. We want the heart to beat as a unit, or actually as several units. We want the atria to fire, all the muscle cells in the atria to fire at the same time, have one smooth contraction, and we want all the muscle cells in the ventricles to contract at the same time and give us one strong push. So the way that that's done, the autonomic nervous system creates an impulse, comes down here, and that signal, okay, so I don't know where the actual pulse comes from, but the main piece of information goes through this synatrial node. It's just a little ellipse of nerve cells, and when it creates this signal, it's going to be propagated through a network of nerves covering the atria, and they're going to go off, but the same signal is going to encounter this atrioventricular node, which is going to delay the signal a little bit so that the ventricle isn't firing at the same time as the atria. It will then carry this signal after the delay through these bigger and then ultimately finer nerves, and these nerves are going to coat the surface of the heart and then provide one synchronous impulse to make the muscles contract, and so they all beat at the same time. And that gives us our nice strong push from the ventricles. Because of this electrical activity, we can actually measure that electrical activity at the surface of the skin, and that's what we're doing with an EKG. So when you actually graph the activity that's seen on an EKG, it looks something like this for each cycle. So the things that are happening here, remember, there's electrical signals for everything. So there's the atria going off, there's the atria recharging, there's the ventricles going off, and then there's the ventricles recharging. And they happen in that order. So the atria aren't that big. So when they depolarize, that's this P peak here, they create this electrical signature. Then they kind of rest for a second, but then they start recharging. Their recharging, their repolarization is often buried in this very high signal that we'll talk about in a sec. But nonetheless, it's happening in this area. It will be represented by a little peak. So when the ventricles are contracting, let's see, they are, this is the depolarization of the ventricles. So this is when the neurons in the muscles are starting to fire. And the actual contraction goes from this point to here. So once that's done, the ventricle is contracted, it's resting, then it actually has to recharge. This is the repolarization peak here, this T peak. I don't have a slide for it, but at this point I just wanted to remind you what systole and diastole were for blood pressure. So systole, we decided in our class we would use the mnemonic S for S. So systole is for the squeeze of the heart, and that's what's giving you the big pressure on your system. So systole is the left ventricle contracting, pushing blood out. This is the highest point of pressure in the system. Diastole is essentially the pressure that remains once the pressure from the ventricle is no longer present. It's actually the inertia of the initial pulse, but it's sufficient to get the blood back to the heart and so forth. And that's the lower number. When you're actually assaying this by taking blood pressure, what you're measuring in the systole is the pressure it takes to actually squirt more blood into the pretty well sealed off arm. And the diastole is when there's no turbulence left. That is when the blood can actually return freely to the rest of the system, so the cuff isn't at a pressure to hold it off anymore. So systole is the squeeze that pushes it out. Diastole is when it can actually come back. Sorry for the diversion there. I will point out that it's important that you're able to actually look at an electrocardiogram, count the distance between peaks, note the distance of time between them, and therefore get the rate and calculate the heart rate from those peaks. So in this case, what we've done is we've looked at the interval of 3 seconds, and in that we see, let's see, 1, 2, 3 intervals, and then some slack both here and here. So what we've decided is that this is 3.75 cycles in 3 seconds. That gives us 1.25 cycles per second. And if we have 60 seconds in a minute, therefore we have 75 cycles. There's different ways to calculate that, but it's all going to come back to looking at the chart and seeing what the rate is.